In this section, we are going to be having a discussion of the conditions associated with inflammation of the meninges. That means we are going to be talking about the cerebrospinal findings in a patient suffering from meningitis. Before we talk about the findings that are associated with the meningitis, let us just remind ourselves of the normal properties of the cerebrospinal fluid. The normal cerebrospinal fluid is an example of a fluid that is going to be clear in its appearance. It is going to be having the presence of cells which are going to be usually only 0 to 5 cells per microliter. We do have the presence of proteins in the normal cerebrospinal fluid in the range of 15 to 40 milligrams per deciliter. We do have the presence of glucose also inside the cerebrospinal fluid, which is in the range of 40 to 70 milligrams per deciliter. And normal CSF is going to be containing chloride, which is around, for the sake of easy remembrance, we say 115 to 120 milliequivalents per liter. As most of us are familiar that the cerebrospinal fluid is going to be having a pressure which is going to vary between 6 centimeters to 15 centimeters of water. Whenever we are going to be having inflammation of the meninges, there is going to be an alteration in the different physical parameters and the chemical parameters of the cerebrospinal fluid. This being a high stuff, we just recall the different findings associated with the different etiological factors associated with meningitis. If we look at a patient who is going to be suffering from bacterial meningitis, patients having bacterial meningitis are going to be having the presence of a turbid CSF fluid on the gross appearance. So it is not going to be a clear fluid, it is going to be a hazy turbid fluid that we are going to be having on the gross appearance. We are going to be having an inflammatory reaction, because of which the number of cells in this condition is going to be increased tremendously. Because of the fact it is a bacterial infection, so the predominant white blood cell that is present in this situation happens to be a neutrophil. Because of increase in permeability, we also expect to find an increase in the amount of proteins present in the cerebrospinal fluid. Now, when you're going to be having more number of organisms like bacteria in the cerebrospinal fluid, for their survival, they are going to be consuming glucose. And for that very reason, what we find is that the glucose levels in this kind of a condition is going to be drastically reduced. Similarly, the chloride levels are also going to be reduced, but mildly. So in contrast to a normal individual, bacterial meningitis is characterized by high pressure inside the cerebrospinal fluid, pressure exceeds tremendously, turbid fluid, increased number of neutrophils, high levels of proteins, reduced levels of glucose and reduced levels of chloride. If we talk about a patient suffering from tubercular meningitis patients are going to be having almost an appearance of the CSF which is characterized by increased amount of fibrin. If you therefore allow this kind of a cerebrospinal fluid sample to settle down in a test tube, it is going to be giving you an appearance which is given the name of cobweb coagulum. Cobweb coagulum. So high levels of fibrin are responsible for giving this appearance cobweb coagulum. If you look at the number of cells, the number of cells in this condition are going to be increased. But what is going to be seen in a patient suffering from tubercular meningitis, these patients are going to be having increased number of neutrophils. They are also going to be having an increase in the number of the mononuclear cells. So in the same individual, you are having an increase in the number of neutrophils as well as lymphocytes or mononuclear cells. This condition is given the name of CSF pleocytosis. Tubercular meningitis patients are going to be having a finding which is given the name of CSF pleocytosis. So as the name is self-explanatory, pleo is plural, more than one type of cell is going to be seen in the CSF in these individuals. That is going to be seen as a finding of a patient suffering from tubercular meningitis. Looking at the other parameters that is seen in these patients, patients suffering from tubercular meningitis are going to be having elevated levels of proteins, but they are not as elevated as you would expect to see in case of standard bacterial meningitis. Glucose levels are going to be reduced. but only mildly. One of the most characteristic findings that we see in a patient suffering from tubercular meningitis happens to be drastically reduced levels of CSF chloride. So this is one of the better ways by which we are actually going to be distinguishing this kind of meningitis from the other causes of meningitis. So gross appearance, cobweb coagulum, there is going to be CSF pleocytosis and the presence of depressed levels of CSF chloride. That's how we arrive at a diagnosis of tubercular meningitis. If we talk about viral meningitis is going to be characterized by clear appearance of the CSF fluid. When we are looking at the number of cells are going to be increased. Just like majority of the other viral infections, the cells which are going to be increased in this situation happens to be the lymphocytes. If we look at the protein levels in this condition are going to be elevated, but not as severely elevated as you would expect in case of bacterial meningitis. The glucose level surprisingly in these patients is going to be nearly normal. We need to be familiar with the fact that there are two important exceptions to this particular rule that we have. So most of the patients suffering from viral meningitis are going to be having nearly normal levels of glucose in the cerebrospinal fluid, with the important exception of the viruses like the mumps virus and the herpes virus. They are the ones which are going to be associated with reduced levels of glucose in the CSF. If we are looking at the chloride levels in case of a patient having viral meningitis, the chloride levels in these patients again is going to be reduced. Talking about the last category of meningitis, which happens to be the fungal meningitis. In fungal meningitis, patients can be having clear appearance of the fluid, or it is going to be maybe mildly turbid. When we are looking at the cells, the number of cells over here is going to be increased. Again, these are going to be mononuclear cells. When we are looking at the protein content, proteins are going to be increased. The glucose levels in these individuals are going to be mildly reduced. And when we talk about the chloride levels, even they are going to be mildly reduced in these patients. So characteristic features that are associated with each of the different types of meningitis would be saliently remembered as bacterial meningitis, characterized by turbid appearance of the CSF, high levels or numbers of neutrophils, very much increased levels of proteins, very depressed levels of glucose, and mildly reduced levels of chloride. When you talk about the bacteria responsible for causing meningitis, classically these can be the bacteria which are something like Streptococcus pneumoniae. We can be having group B streptococci like Streptococci agalacti. Meningitis in children can also be seen apart from group B streptococci caused by Escherichia coli, and we can be having meningitis caused because of the gram-negative diplococci like Neisseria meningitidis. When we are talking about viral meningitis, classically seen because of enterovirus infection, which is being transmitted through the fecal-oral route. Viral meningitis is characterized by the presence of nearly normal levels of glucose in the patients. When we are talking about tubercular meningitis, characterized by cobweb appearance of the CSF, and very characteristic finding, you're going to be having severely depressed levels of CSF chloride in these patients. These are the different parameters which are useful for distinguishing between the various factors associated with meningitis.